Thank you for your interest in this poster presentation. I am Vaibhav Mehta, a senior scientist at Altasciences Laval, and it is my pleasure to present this compilation of work prepared for the Oligonucleotide Therapeutics Society 2024 conference in Montreal. This presentation titled Strategies to Improve Assay Sensitivity for Bioanalysis of Therapeutic Oligonucleotides through Ligand Binding Assays highlights 3 case studies where varying assay optimization strategies resulted in superior assay sensitivities compared to conventional ligand binding assays. Hybridization-based ligand binding assays are the preferred bioanalytical platforms from discovery to the clinical phases owing to their inherently higher sensitivities and lower operational costs. As depicted in Figure 1 and 2, typical ligand binding assays for oligonucleotide analytes implement either a single probe nucleus-dependent approach or a dual probe approach. Both approaches are applicable onto the conventional formats of enzyme-linked immunosorbent assays, ELISAs, or electrochemiluminescence ECL-based assays. While the single probe approach utilizes nucleases specific to single-stranded DNA, such as S1 nuclease or micrococcal nuclease, to eliminate unhybridized probe excess or analyte-free probes, the dual probe approach simply utilizes the requirement of the analyte-bound capture probe for recruitment of the detection probe. Detection in ELISA is mediated through fluorescence generated through an enzyme redox reaction, such as the alkaline phosphatase enzyme, and detection ECL assay is measured through luminescence generated from electrochemically activated luminophores, such as ruthenium. There is an increasing demand for higher assay sensitivities to support deeper insight into the pharmacokinetic profiles for preclinical applications and to support quantitation of potent compounds dosed at lower concentrations in clinical applications. This work showcases 3 bioanalytical methods where higher sensitivities were achieved by assessing different reading platforms, probe designs, and signal amplification strategies. In the first method, Case 1, a previously validated S1 nuclease-based ELISA for the quantitation of an antisense oligonucleotide, ASO, in monkey serum was to be transferred for use in rabbit serum. As seen from Table 1 and Figure 3, the method in monkey serum has a sensitivity that is a lower limit of quantitation at 1 nanogram per ml with a signal-to-noise of about 2.6-fold at that level. With an upper limit of quantitation at 100 nanogram per ml, the dynamic range of the method is 100-fold, and the signal-to-noise ratio at the upper end is approximately at 110-fold. The method performed poorly upon a preliminary transfer into rabbit serum with a lower signal-to-noise ratio. Therefore, several changes were implemented to improve performance at the lower end, primarily involving switching of the S1 nuclease with micrococcal nuclease and switching the detection format to electrochemiluminescence. As represented here in Table 2, the updated method has an improved sensitivity at 0.5 nanogram per ml with a superior signal-to-noise ratio of about 6-fold. Additionally, with a higher upper limit of quantitation of 200 nanogram per ml and 400-fold dynamic range, the signal-to-noise increased 18-fold compared to the previous method at the upper end. These improvements are evident from the linearity of the regressed curve. Precision and accuracy of the updated method was successfully demonstrated in 7 runs, as seen in Table 3. Switching nuclease alters the interference coming from the metabolites. Table 4 demonstrates the overall improvement in the interferences coming from the N-2 and N-4 5' end metabolites from ELISA to the ECLI method. Additionally, these changes overall reduced the workflow by 40% and reduced costs by 60%. Moving on to the second method, Case 2, a method was being developed de novo for the quantitation of an antibody phosphorodiamidate morpholino PMO conjugate in mouse tissues. Therefore, during development, the dual probe format and single probe nucleus-dependent format on ELISA and ECL detection platforms were explored using the conjugated PMO payload. This is depicted in Figure 5, and the data from that is shown in Table 5. This preliminary testing revealed a significantly superior signal-to-noise ratio from the micrococcal nucleus-dependent single-probe ELISA format, which was chosen for further optimization with the conjugated payload. Now moving on to Figure 6 and the data represented in Table 6. The conjugated payload has considerably lower instrument signals at the same concentrations as the free payload, indicating that the antibody interferes with the detection. Therefore, protease treatments with trypsin and proteinase K were further explored. The data presented showcases the performance from trypsin treatment, as that was a better fit overall versus proteinase K in context of not only performance, but also running costs. As seen from Table 6, a 10-fold improvement in sensitivity is obtained with trypsin treatment. The method proved reliable in quantifying the conjugated payload between several different tissue types, as highlighted in Table 7, and this further demonstrates its robustness. Finally, in the third method, Case 3, a qualified micrococcal nucleus-dependent ECL assay required re-optimization for higher sensitivity for quantitation in mouse tissues and prospective clinical studies in biopsied human tissues. The current method performed with a sensitivity of 1 nanogram per ml with a signal-to-noise ratio of about 6-fold. With an upper quantitation limit of 200 nanogram per ml, the dynamic range of the method was about 200-fold. The method was re-optimized to a dual probe format, where signal amplification was pursued through labeling of the detection probe with the AU dejoxygenin tail. The AU dejoxygenin tailing was achieved through terminal transferase, and different incubation durations typically led to varying tail lengths. Table 10 highlights from that optimization, where the 30-minute tailing procedure provided the highest signal-to-noise ratio at the lower end. Overall, the improved dual probe ECLIA has a sensitivity of 0.01 nanogram per ml, a 100-fold improvement from the previous method, and a superior 1,000-fold dynamic range. In conclusion, these cases highlight some of the strategies through which quantitation of therapeutic oligonucleotides by conventional ligand binding assays can further be improved. These improvements not only provide methods with higher sensitivities, but also prove to be robust, cost-effective, and time-efficient. With that, I would like to thank my supervisors and all other Altasciences employees that were involved in these studies.